Welcome to the biochemical story. Today we will be discussing the mechanistic steps of the TCA cycle, or the citric acid cycle. The reactions of the TCA cycle follow a circular path, starting from oxaloacetate and acetyl-CoA. The first reaction uses citrate synthase as the enzyme and forms citric acid. Second reaction, we have aconitase. Third, isocitrate dehydrogenase. Fourth, alpha-ketoglutarate dehydrogenase. 5, succinyl-CoA synthetase. 6, succinate dehydrogenase. 7, fumarase. And 8, malate dehydrogenase. Here we'll start with oxaloacetate and acetyl-CoA in the first reaction. First, an aspartate residue is protonated by hydrogen on the acetyl-CoA. The electrons then attack the carbonyl on the oxaloacetate and opens up and becomes protonated to an alcohol. We then form this intermediate with oxaloacetate and coenzyme A. Our base picks a proton off of a water molecule and the electrons attack the carbonyl on the intermediate. The electrons go up onto the oxygen, back down, and we kick off the coenzyme A, where it's protonated by an acid. We then form citric acid. In step 2, citric acid is interconverted to its isomer through the enzyme aconitase. Citric acid is deprotonated and a dehydration reaction occurs where water leaves, forming the intermediate cis-aconitate. The next step is where a base picks proton off of water and the electron attacks the double bond in cis-aconitate, opening up for it to be protonated. In step 2, after the rearrangement of the water, we form isocitrate. In step 3, we start with isocitrate and form alpha-ketoglutarate through the intermediate oxalosuccinate. The alcohol on isocitrate is oxidized to a ketone and the hydrogen leaves and reduces NAD+ to NADH. The intermediate oxalosuccinate is then formed. An oxidative decarboxylation reaction occurs, releasing a CO2, forming alpha-ketoglutarate. In step 4, another oxidative decarboxylation reaction occurs. A CO2 is released, and a new bond forms between the alpha-ketoglutarate and the coenzyme A, and the hydrogen reduces NAD+ to NADH. In this step, a CO2 is released, coenzyme A is bonded to the former alpha-ketoglutarate to form succinyl-CoA, and an NAD+ is reduced to NADH. In step 5, we start with succinyl-CoA and use the enzyme succinyl-CoA synthetase. We have an inorganic phosphate, a base picks a proton off, and the electrons on the oxygen attack the carbonyl on succinyl-CoA, and coenzyme A is kicked off. We then form this intermediate. The phosphate group is not directly transferred from the intermediate to GDP. Instead, a histidine residue from succinyl-CoA synthetase facilitates the transfer, where an electron from the histidine residue attacks the phosphorus in the phosphate group and opens up the bond. Then the phosphate group on the histidine residue is transferred to GDP, forming GTP. In this reaction, we go from succinyl-CoA to succinate. GTP is later converted to ATP. In step 6, we have succinate and succinate dehydrogenase as the enzyme, and we form fumarate. FAD is then reduced to FADH2. For the dehydrogenation reaction, we start with a base, picks off the proton from succinate, and a double bond is formed, and the hydrogen goes over and reduces the FAD to FADH2. In step 7, we start with fumarate, and for the hydration reaction, we use fumarase to convert to malate. A base picks a proton off of water, and the electrons open up the double bond, and an alcohol is added. In step 8, we start with malate, and we have NAD+, which is reduced to NADH. The alcohol is oxidized to a ketone, and the proton is kicked off and reduces NAD+ to NADH. And this is our third NAD+ that we've reduced to NADH in one turn of the TCA cycle. We then form oxaloacetate, right where the cycle started. In one turn, we reduce 3 NAD pluses to NADH on step 3, step 4, and step 8. 1 FAD is reduced to 1 FADH2 in step 6. And we phosphorylate GDP to GTP. We also release 2 CO2 molecules. In the TCA cycle, we use 8 different enzymes: citrate synthase, aconitase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinyl-CoA synthetase, succinate dehydrogenase, fumarase, and malate dehydrogenase. I hope this video helped. Thanks for watching the biochemical story.